stars. And we have 4 rock stars here to present to you the work that they do every day to make a difference in what's happening, not only at Jefferson, but in the world. I'm going to introduce our speakers in order in which they will speak. And rather than me come back onto screen and reintroduce, we will just go from the first speaker to the second speaker, third and fourth. Our first speaker will be Dr. Mark Tikaczynski. He is our Provost and Executive Vice President for Academic Affairs, and he is the Anthony and Gertrude De Palma Dean of Thomas Jefferson University. Next, we will hear from Dr. Matthias Schnell, who is the Professor and Chair of Microbiology and Immunology. He is an inventor of many vaccines, most notably the subject of today's conversation, which is CoraVax, one of the coronavirus vaccines under development. Dr. Heather Rose is a great colleague and a brilliantly talented person who not only understands the science, but understands how you put deals together. She is Vice President of Technology, Licensing, and Startups. And batting cleanup is Dr. Rose Ritz, the Executive Vice President of Innovation, our Chief Innovation Officer, and one of the best deal makers. Rose flies below the radar, but boy, does she make things happen. So I'm delighted to introduce you to this panel, leading off with our Provost and Dean, Dr. Mark Tikaczynski, an innovator in every way in his own right, and he will open today's event. Thank you so much for being here, everyone.
Thanks so much, Donna, and really glad to be here and doing the intro for Matthias Schnell, one of our absolute stars. I don't know if we're going to get the slides up here.
Let's start slide sharing, yes.
Terrific. Next slide.
Next, yeah.
Terrific. So I just wanted to spend a couple of minutes in the lead in here to put the work that we're doing at Jefferson into a context. We've been on a trajectory over the decade plus that I've been here at Jefferson as we take advantage of a world and continue to build a world-class research faculty. Team science and interdisciplinary interchange have been high on our priority list. Really pushing towards this idea of programmatic science, and then a comprehensive state-of-the-art shared resources. And in that decade, we've actually more than doubled our extramural funding at the university. A big part of driving that has been this idea of taking labs, clustering them into programs, examples being tumor environments, synaptic biology, and so on. One of them is emerging infections. And now wrapping them into even a larger wrapper, which would take tumor environment as part of, let's say, cancer. But particularly for today's talk, the emerging infections expertise to pandemic preparedness. And so one of our areas of focus strength at Jefferson now is immunity to emerging infections primarily within the Department of Microbiology and Immunology that Matthias Schnell leads. And we now are transitioning some of those programmatic themes into real centers of excellence. And so hence the Jefferson Vaccine Center. Next slide, please. And so we now have a Jefferson Vaccine Center, but what we're starting to put together and will launch undoubtedly within the next couple of months is a more encompassing concept of a Jefferson Center for Vaccines and Pandemic Preparedness. How do we start to mobilize the resources and put them in place so that we're not starting from ground one? And the idea is not simply to do it as a Jefferson-only exercise, but how do we create a consortium of partners, both within the country, nationally, but also internationally, where we would start to assemble the reagents, the genetic vectors, the assays, and the tools that one would need that might anticipate where potentially that next pandemic would come from. Not simply just a variant of coronavirus, which it could be, but entirely other viruses that are out there. And so In that evolution, that now is also part of a much even larger platform, which is the concept of a Jefferson Center for Global Health Security. As we inventory what we have here at this university, there are a number of elements that are starting to come together. We recently launched the Jefferson Institute for Bioprocessing, and we have emerged as a significant location for training of the workforce that will be driving biologic production, which is a bottleneck in pharma these days. We launched a computational medicine center, so our capabilities in data sciences, specifically as it relates to computational biology, mining large bio data sets, really has gone to a next level, and that can be deployed in the context of the vaccine development. But thinking about pandemic preparedness, global health security gets you thinking about spaces and architecture. And within our College of Architecture and Built Environment, we have launched an Institute for Smart and Healthy Cities. And then specifically on the global side, we have focused our energies in a number of countries, really in a handful of countries where we go deep: India, Israel, Ireland, Italy, Japan. And these are countries where we see collaborating around this Center for Vaccine and Pandemic Preparedness, as well as global health security. And among our 10 colleges, there are a number of colleges that really fit in beautifully and have existing programs that we'll be able to draw on, even in areas like design, engineering, and commerce. Philadelphia University was originally founded as a college of textiles. And so the area of smart textiles, textiles that can be deployed towards health and anti-infectives, as just one example of what we can be doing. Next slide. And I just wanted to highlight here, because we're pretty excited about this these days. In the past year, we launched this Jefferson Institute for Bioprocessing that I mentioned. We started off focusing on protein pharmaceuticals and training the workforce for protein pharmaceutical production and biologics. But we now are doing a significant further investment in expanding our space in Springhouse and the Innovation Park there, where we'll now also embrace training the workforce for cell and gene therapy and, importantly, advanced vaccine production and development. I think we can be really proud of what's going on in the Philadelphia area in general as we emerge as a significant locus for cell and gene therapy work, but also in terms of vaccine production. So I'd like to now immediately turn it over to Matthias Schnell. Matthias is the founding director of the Jefferson Vaccine Center. He comes across as quite modest, but the reality is that he is viewed internationally. I can verify that. I've seen that as really one of the fathers, in many senses, of viral vaccines, and has really pioneered some key platforms for viral vaccine usage in a whole array of different emerging infections. So, Matthias, the floor is yours.
Thank you very much for this nice introduction. Can I have my first slide, please? So just let me introduce, you heard about me, but what is actually more important is a team we have here at Jefferson who does actually some practical work. So so most senior, and I list the people by seniority when they join the lab. Chris Werbley, he's here for a long time. He's really keen the lab. He clones and makes basically every vaccine construct we need. So we use mostly rabies virus as a vector, but we also use measles or vesicular stomatitis virus. And he developed vaccine process. He helps with also biochemical assays. And in addition, he also uses and works on these neurotracer viruses, which are used for brain imaging, which I can't talk about today. So Trisha, she works on the rabies-based vector against the emergent infectious disease. So she switched mostly now to SARS coronavirus 2. But she also works in parallel on the measles-based vector against Zika virus. Remember, Zika virus, nobody talks about it anymore, causes huge burst effect. it's also important vaccines. And I have some PhD student, Catherine, works on the Ebola vaccine, also a vaccine which we urgently need and looks in functional analysis of different adjuvants. Gabby works on Crimean Congo hemorrhagic fever virus, another emergent infectious disease. She also helps with our Lassa virus vaccine. And also SARS coronavirus, which basically everybody works on. Then I have Chantel, we just got new funding for grant for R1 in collaboration with the University of Maryland with the Borrelia vaccine. You may know that about 500,000 people get infected with Borrelia in the US every year. Is still a huge problem, not as high as SARS. And so on. We work on other Lassa virus vaccines and so on and so on. Can I have the next slide, please? Oh, this is a picture we took yesterday about the people. Chris took it, so here you can see the whole team working on all these vaccines. Next slide, please. So this just gives you an overview on what we work and what we think is important to work on. And after that, I will switch exclusively to SARS coronavirus 2, which is on everybody's mind. But you may remember botulinum neurotoxin and anthrax was a huge issue as biodefense vaccines. We have them in the freezer in the preclinical development. We don't work actively anymore because there's not a big interest. HIV certainly an important vaccine. Then Nipah and Hendra, emerging viruses in India, also spread by aerosol, are lethal 50%. So that's big concern. Ebola virus, and so more in the where we move in the clinic soon in a phase 1 is Sudan virus. That's done with a contract with NIH. Lassa virus, which is a huge problem in West Africa, and certainly the SARS coronavirus too. But all these vaccines listed here are of importance, and we could continue, but there's only so many we can do. But that's all actually diseases which caused problem during the last 10 years. We just keep on forgetting about it. Next slide, please. So now I switch completely to our SARS corona, our COVID vaccines. And just to tell you a little bit what is out there, we have the protein-based vaccines, which are used on, protein subunits. Basically, you produce a spike protein, which is at the surface of the virus, and use that to immunize, also viral-like particle. That's a little bit in the direction our vaccine goes. Both of them actually didn't reach an approved vaccine yet. What got further developed and what you heard most of it as the RNA vaccine and DNA vaccine, whereas actually also DNA vaccine didn't result in any approved vaccine yet, but the RNA vaccines, the Moderna and Pfizer, that's a leading candidate. They basically based on a lipid platform which contains RNA encoding the SARS corona spike protein, and then that is injected, fused to cell, and the protein is expressed. These vaccines are highly efficient, have also quite some side effects, but are the 2 approved vaccine in the US, in addition with the so-called Johnson & Johnson vaccine. We can see that on the next slide, please. Okay, here you see the virus vaccine. There is the weakened virus, which is probably never coming to market. It's actually attenuated SARS coronavirus, but the inactivated virus that's actually used in India and China. They tried to get to the US market, but it's not approved yet. And then more, the Johnson & Johnson vaccine is a viral vector vaccine, which is based on a non-replicating viral vector, and it's adenovirus. And basically you take an adenovirus and you put in part of coronavirus 2, and then you immunize with that virus, and that expresses a viral protein and you get an immune response. This vaccine, the Johnson & Johnson, is a little bit less efficient than the RNA vaccine, about 70%, but it's a single shot vaccine. So the problem with this viral vector vaccine can be that, a second or third application will be difficult because you develop antibody against the viral vector too. All right, next slide. So this shows what is out there. We have 40 in phase 1, 27 in phase 2, and so on and so on. So there are many, many vaccines currently out there. So you may wonder, why do we need more? And my point is, what is currently out there is all pretty much based on the same platform. So you have adenoviruses, you have RNA vaccine. If at some point they don't work anymore based on side effects or immune response against the vector, we have really not a lot left. And as you know, we have now new mutants coming. So as more as happier at this point. I think right now we have about 1% of the world population vaccinated, which is really not a lot. Next slide. So this is basically our approach. It's a rabies-based vaccine, which we call CoraVax. Actually, the name created was by Mark. And basically what we use is ability for rabies virus to include foreign protein. And as you know, rabies virus is a very well-established vaccine used in hundreds of millions of people over the years. So what we genetically manipulate the rabies virus and put in a gene encoding part of the spike 1 protein. That protein is incorporated, and then we vaccinate instead of with a regular rabies virus vaccine with a CoraVax vaccine. And we know that we get really nice antibody responses against rabies virus with a rabies vaccine, and we were able that we get similar good responses against SARS coronavirus. And this certainly will be considered a very safe vaccine because it's a deactivated vaccine. So it's basically a dead vaccine, which a lot of experience in its use, including children and pregnant women. Right now, we actually have no vaccine approved for the under 18-year-olds. We also have to consider immunosuppressed people. Next slide, please. So this is just why we started it and why we like rabies so much as a vaccine platform. Even the name rabies itself sounds scary, but because it's such a scary disease, we have really excellent vaccines, and that's since a long time, going back to Pasteur, which, we talk about 100 years ago. One of the first developed vaccine was one against rabies together with a smallpox vaccine. So long-term protection, and that's really not considered right now because we just don't know. Rabies virus vaccine often provides lifelong protection. For example, I was vaccinated against rabies 30 years ago and have still very, very strong antibody against rabies. We convinced that it will be safe for all population groups because we have historical data that this vaccine have been used in children and pregnant women without any issues. We showed before that this vaccine works well on different platforms. The most related virus was the MERS coronavirus. You remember that in the Middle East, a huge problem, actually way more deadlier coronavirus kills about 50% of the people infected. We think it's easy to produce and to reconstitute. It has a long shelf life. It doesn't need this crazy -60 temperature. It's actually stable at 4 degrees and at room temperature for quite some time. There are a lot of manufacturers out there which can produce rabies virus vaccine. So we should be able to scale that up, which is also important. And we actually work with one of the largest vaccine production company worldwide, which is Biotech, which already produces rabies vaccine for the developing world in large amount. And actually the developing world is the one which suffers most on the not availability of this vaccine. And they also can't afford this RNA vaccine, which are quite pricey. If an inoculation costs your yearly income, that's probably not a go for a lot of people. Next slide, please. Now I want to show you some, data on our vaccine. So what we and that also I want to show, because that's actually how you develop a vaccine. You make your vaccine, you produce it, and then you test it first in mice. We did test it in a prime boost approach. You heard a lot about that. Most of these new RNA-based vaccine are also prime boost. And we also tested the live, where we certainly don't consider the live for humans. And as you see, it was day 0 and 21, and then in B, you see how the antibody responses increases over time. Basically, if it goes up, that's a good sign. So the blue bars go up over time. Can I have the next slide, which shows you the most important point? And what is actually most important, and it's getting more and more prominent, this knowledge, is the virus neutralizing antibody titers actually tell you protection. This goes so far that from the titers detected in human trials, they already have models to calculate the efficacy. And they did it. I just heard a talk on that today. And what you see here, and the hallmark of most vaccine is that they're at least as good as convalescent sera. And I'm not sure about the background of all the people. Anyhow, convalescent sera is for people who recovered from the natural COVID infection. That gives you a baseline. And then we also have an acute infection here listed. That's normally you have higher antibodies titer during that. And what you can see is CoraVax in a prime boost. An adjuvanted gives us neutralizing titer 1 to 600, and adjuvanted 1 to 2,200, which is about, 5-fold higher than acute infection and about 20-fold higher than, or even more than, a convalescent sera. So we way above what we need, which is encouraging. In addition, we also get nice antibody against rabies virus. And if you know that rabies virus kills about 60,000 people every year, mostly children, this would be a great, additional benefit from a vaccine like that. Basically cheap, stable, and against 2 diseases. That's what the third world looks for. Next slide. So immunization and pre-existing immunity, I'm really worried about viral vector and pre-existing immunity, because if you use a viral vector, you get this immunity against the vector, but ours is a killed vaccine, so it doesn't have to enter a cell. So we did a quick experiment for that. We used in group A one unrelated vaccine, and in group B, we just used the CoraVax, and then we did the immunization. So group A, in the presence of pre-existing immunity against rabies, and in the other groups without it. Next slide. And what you can see actually is that we don't have any significant difference with a prior vaccination against rabies virus. So this is probably a platform which can be used multiple times, which would be a big advantage. And so rabies immune response, you can see, is also equal. So at some point it stops going up, which is also important. Next slide, please. So long-term, also you hear a lot about this vaccine and how terrific they are, but we really don't know for how long they are terrific. Adenovirus immune responses go away quite quickly because you get infected by basically yearly. So that could be a problem if the vector dictates the immune response against the expressed antigen. That could be a problem. So we actually did 2 groups. We have unvaccinated and our vaccinated animals shown here. And then we looked in the long run in the next slide. This is long-lived plasma cells. I think that was 6 months after infection. So basically you pull out the cells out of the bone marrow of these mice and you see very nice immune responses. These are antibody secreting cells. And we know now up to 9 months, SARS is not around that long. We know that we have at least 9 months in mice, and a year of a human is, I think, 7 days of a mice. So we would expect that we have really long-term immunity here. And that's our preliminary readout. As I said, it's all mouse data, so we have to go in mice, but in humans, but that will happen soon. Next slide, please. And now I show you, it's the last part, vaccine in hamster. Hamster, I think, in my opinion, in very, very personal opinion, is the best model actually for this vaccine. So there's a lot study done in non-human primate. They don't get as sick as hamster gets. So I think the hamster model is actually better. Next slide. What we did, we actually did 2 inoculation, works great, but then we pushed it a little bit and look, can we get protection with a single immunization with our adjuvanted vaccine? And we use actually the adjuvant is squalene, which is similar to MF59, which is a well-established adjuvant without any problems used in many vaccines. So you see the design, we immunize at day 0, and then we follow this animals' immune response 14-18, then we challenge them with SARS coronavirus 2. It's the Washington strain, that's the first strain which was discovered. And then we have to sacrifice this animal over time at day 42, 46, and 53, because hamster as humans actually don't die, they get sick, but humans barely die, but can be quite sick. So this is the experiment. Next slide. That shows us the immune responses at day, and just look at the right side. Day 0, the red curve is actually controlled sera, a positive control. Day 14, day 28. What fascinates me about the rabies vaccines, the immune response is just going up, up, up. Even after 20 days, you see an increase after 14 days. So it seems to stick around for some time. Next slide. That shows us the challenge data. You see day 3 post-challenge. So immune response went a little bit up at day 7 dramatically, and as day 14, which means we have a nice effect. The vaccine actually stimulates your immune system, creates a memory response, and if the SARS coronavirus comes in, it reacts quickly and increases the antibody response dramatically. Next slide. And that actually shows you the immune response compared to animals which survived the infection without vaccine. That's a black bars. So as I told you before, these animals actually get sick, but they survive, similar to human. But you see the immune response of the surviving animals is really quite, that's a triangle, the black triangle is about, a factor 1,000 under what we see in our animals. So the vaccination definitely increases, and at day 28, we have already higher immune responses than you see after cured reconvalescent hamster sera. So that's all in favor of this vaccine. Next slide. And that shows you the protection against weight loss. You have a little dip, the blue line is a vaccinated one, so black wire is an animal which were not vaccinated. You see a little drop, so they get a little bit sick up to day 3, but then they rapidly recover. I think you agree with me, if you get COVID and you will be only sick for 1-2 days. That's almost as good as if you get an RNA vaccine, which also makes you sick. So, and it goes up and it's highly significant. Next slide. Now I show you some histology. I really like that. It was because it really shows you the disease. So you see 3 days post-challenge. These are lungs from all the animals we actually challenged and which got the vaccine. You see a little bit inflammation in this, a little bit blood, but in general, not a lot of antigen. You see that to the right, the brown stuff, that's a viral antigen. So we have initially infection as expected, but then the virus is rapidly cleared. So keep that slide in mind when we step to day 3 for the non-vaccinated animals. Next slide, please. Okay, and that's how it looks if you get infected and were not vaccinated. And you can tell all that brown, which is labeled SARS coronavirus 2 end staining, that's all virus antigen. So it's basically looks like 20 to 50% of the lungs are infected. You also see a lot of inflammation and blood, and that's what causes actually the disease in human too. Next slide. I think we have 2 more, then I should be done. That shows it at 7 days. You still have a little bit inflammation in our animals, but not too much. The antigen is almost gone. And if you go to the non-vaccinated animals in the next slide, you see, I mean, even if you're not a pathologist, all that dark red is actually bleeding. And the pathologist also saw later on some buildup of fibroblasts. So this is, even when the virus is cleared, you have this almost immunopathology by delayed immune responses. And you can see the SARS coronavirus 2 staining is almost gone, but you still have this huge, basically overreaction of the immune system, or the wrong immune response. Whatever it is, that's still debated. Next slide. And that shows you what the pathologist says after evaluation. So it was at Jefferson, a lung pathologist, which does that on a daily basis. And you see the score is basically 16 is as bad as it gets. At day 3, you see we have great reduced pathology from almost nothing to 12. But remember, this is a single inoculation. With 2, it may got further reduced, but it's pretty good. And at day 7, the pathology is almost gone, whereas the non-vaccinated one have really still a high pathology score. And at day 14, we have mild diseases in 3 animals, and 2 were completely cured, whereas the other one don't look so great. And that's actually when you get this fibroblast, and it's probably that you can get that long-term damage of the lung. Next slide. So what do we think? We think that our vaccine is highly immunogenic in mice, highly effective in the hamster SARS challenge model, quickly induces a generation of neutralizing antibody. And as I said, in the meantime, that's believed to indicate protection. It protects the animals, promotes viral clearance, and reduces the viral load in the lungs to 0. I didn't show this data, but at day 3 at another experiment, there was no virus to be isolated anymore. Neutralizing antibody, Th1 bias or balanced Th1 responses, demonstrated prolonged immunity to SARS coronavirus, 56 days. That's actually outdated. We now at 180 or 275. And viral clearance not only in the lungs, but also in the nasal turbinates, which I also didn't show, but that's important for transmission. So we think we have, at least after 3 days, a transmission blocking vaccine. And 1 dose actually works. We may still go for 2, but at least 1 dose gives already very good protection. Next slide. That just gives you who I have to thank and who I'm really grateful. All the people in the lab, I showed that before. Mauk, which is a great advocate. Heather, which works really hard on that. As does Rose Ritz, which also is, set our head right if we get a little bit too excited. That's helpful. And then from pathology, all this, I'm really, really happy that they did that. Pambos did that for me and gave this report. We got funding from the vaccine center, from NIH, from Bharat, our Indian collaborator, and I should mention the Commonwealth of Pennsylvania gave us also a grant, and that was really kind of all supported and advocated by Jefferson. So thank you very much, and that ends my talk.
Thanks, Matthias. Heather, you're up next.
Great, thank you so much. Great transition, I think, to me with the provost background on Jefferson with vaccines, and then Dr. Schnell getting deep into the development of CoraVax. And now I'm going to talk about CoraVax from the innovation side in regards to deals in the translational development. I'm going to start by talking about my team and what we do, and then I'll transition over to the CoraVax story and the translational development of CoraVax and partnering around CoraVax. So I am myself, by background, I'm a molecular biologist / virologist. Worked got my PhD at Penn in gene therapy using viruses as gene therapy delivery vectors. So luckily in this instance had some on-point background, not always the case in regards to the stuff I work with. This is definitely you get what you get and you don't get upset situation in regards to what I deal with and comes my way. But this one was particularly interesting to me given my background. After I finished my PhD, I went to law school and worked at GSK's corporate IP department while I was in law school. I'm a patent attorney by education, but pretty much right after that, I started working at Drexel University, managing their life sciences development projects in regards to patents and partnering. And then I came to Jefferson in 2017. In my current role at Jefferson, I have a great team that I really do believe I need to give a tremendous amount of credit to for how much they support everything that I am capable of achieving. So I would like to just quickly point out that our office in every function that we do in regards to patents, licensing, marketing, and contracts is a team lift. And so I'd like to take a minute and thank Robert Arcadipane, who patent juggles a million other things and helps me catch the light bulbs before they hit the ground in every aspect, I would say, of what I do. Sharmini Essen, who's our marketing maverick, she helps us make strategic marketing plans and put together marketing materials. Beverly Harris, who's our contract specialist and reads and reviews all the documents related to materials that are coming in and out of TJU and all the associated intellectual property clauses and ramifications. And Dr. John Bohue, who manages licensing matters and helps review incoming IP and outgoing IP and associated deals along with me. So they are all very critical to what I do, and it would be disingenuous of me not to afford them the credit for all their support. Together, we manage over 300 active patents, and we do about more than 100 contracts a year of one sort or another with outside partners to Jefferson that relate to the intellectual property that we're shepherding through this process. So I was on a family vacation in Chicago visiting my brother when the world became aware of SARS-CoV-2. I had already met with Dr. Schnell at that point and knew he had a vaccine quite far along in development. And he had been honest with me that he was very concerned about it spreading to the United States and becoming a global pandemic, and that it looked worrisome. And I distinctly recall flying home and seeing people on my plane wearing masks for the first time. And that was the first time that was becoming normalized in public, and that was early March 2020. During that same trip, I was slipping away from my family functions to help get the CoraVax patent filed. And before I left Chicago, it was in fact filed. And pretty much the instant that it was filed, Rose and I started reaching out to potential partners because of the very grave matter of coronavirus spreading rapidly around the globe, and how clear it was to all of the vaccine people in vaccine development that we needed not only a solution, but kind of all hands on deck for a solution and maybe multiple solutions for testing and vaccines. I sent out feelers to at least 20 companies, if not more, and I'm sure Rose did the same, and as did other people at Jefferson. Actually, we had other people at Jefferson sending us leads of companies at large pharmaceutical companies that were interested in meeting with us and talking with us. Rose and I took a lot of meetings, and it became clear to us that for many of them, they already had a horse in their race, their own vaccines in development, and it was a tire-kicking expedition. Or they might be interested in ours at some point. So they were keeping their ear to the ground to see what else was in development should whatever they had put up first fail, right? However, with Bharat, the conversations were moving along very well. And on our end, we saw them as an absolutely fantastic partner for this project because they are the world's leading manufacturer of rabies virus vaccine, in addition to being a World Health Organization pre-qualified vaccine manufacturer, which means that the World Health Organization has verified that they consistently produce vaccines of a quality that they can be trusted as a vaccine manufacturer. So they are very well known around the world, and in India, they produce a number of vaccines for pandemic diseases, rotavirus, polio, rabies, and that's just a few. And they also had the appropriate manufacturing facilities to start producing CoraVax along with us quite quickly. And it was really just like grease lightning, how well it went putting in place our arrangement with Bharat and finishing up our preclinical package of development and getting to where we are now pretty fast for university timeline, or really a timeline anywhere. And I also want to take a moment and thank Donna and her team, because the Commonwealth grant that Dr. Schnell referred to was super critical to us too, and came just in time. She brought in $500,000 of funding from the state that helped assist getting this project into complete development and the place we are now, which is preparing for TJU to move this into humans. So I have one slide, and I wanted to just use it to walk through the timeline to impress how fast this moved and how rapidly we were reacting to what was going on around the world. So Dr. Schnell's lab had already produced CoraVax by January of 2020, and I had met with him. And actually, I remember he had a board in his office, and he was mapping out each thing along the way, and certain rewards and incentivizing people to stick around and work hard on it, which I'm sure has been a challenge in that lab. We filed the patent on March 6th, and at that point, almost concomitantly, we started using the vaccine in animals to prove that it worked the way that we thought it would in raising antibodies against SARS-CoV-2 and in killing SARS-CoV-2 or inactivating SARS-CoV-2. So the first was just, am I getting the antibody reaction that I want? And then the second was, is it doing the thing I want, right? You could have the antibody reaction and unfortunately not see the inactivation. So those are the next two things that happened right out of February through March and April. Then we did the Bharat deal in April. So this is really happening just like this. The Bharat closed in April, and we had GMP CoraVax, or good manufacturing practices, basically a quality product of CoraVax that theoretically could go into humans pending regulatory approval by around the end of June. All of the rest of this just shows the different safety testing that CoraVax went through to get to the point where we are now, which is submission for regulatory documents in the United States. When the calendar on the bottom switches over into yellow, this is what we're currently working on, which is preparing a pre-IND filing in the United States, and Bharat is also on their own timeframe approaching human studies. That's pretty much it. And I do just want to take a time to thank everybody on the panel, Dr. Tikachinsky, Dr. Schnell, Donna and her team with assisting with the state funding, and Dr. Ritz. This really, really was a group lift. And good job, everybody. And here we are. Let's keep going. Not time to rest yet.
You can go to the next slide. Thanks. Hi, folks. It's Rose Ritz. Good to see you all. Thanks for joining us. I wanted to give a word on vaccine manufacturing. So much of what we've heard about is the Jefferson side of the equation, because we're all Jefferson people. But there's some things that are different about vaccines than other sort of drugs that are made. And I wanted to just highlight those for a moment and say this, that when the regulatory authorities approve a vaccine, they're not just approving the specific biological entity, the chemical formulation, which often happens for sorts of drugs. What they actually approve is the process by which that thing is produced, tested, and released for use. And what you see in this picture is the usual sort of process of making a vaccine, where the company, the Bharat in this case, or some other partner, the person who's making the thing, has to develop a process and test it and scale it up. And they have what they call the phase 1 production, the phase 2 production. They validate. There's all sorts of work that's going on the manufacturing end that is not easy, obvious, or easily or easy to figure out. And very subtle changes in the production process. You're making these things in huge vats and huge facilities. They're like football fields, and it's actually a beautiful facility in India. Bharat has sent us pictures of the facility. It's a kind of remote and beautiful location. Very subtle changes in the production process can alter the final product. Very small things, some little impurity can change the efficacy or the safety of your downstream product. It's really tough. And something interesting, I think, for those of us who work in patents and in company formation, is that patents around vaccines, when you have vaccine patents, they often protect this manufacturing process along with the antigen, the work, the science work that the science teams do. There are patents that come up around the manufacturing processes for vaccines. And sometimes those process patents present a more difficult, what we call a barrier to entry, than the patent around the material itself. And that's all very different than other sorts of drugs where the material itself is the core of the patent estate. Next slide, please. So here I've repeated one of Dr. Schnell's slides, but I want to jump right here to number 4, this whole notion of the logic behind CoraVax, cost-effective manufacturing infrastructure in place. This goes by so quickly, but I want to emphasize that there are 20 manufacturing plants around the world already today that are manufacturing the rabies vaccine, which is the core underlying vaccine of CoraVax, churning out millions and millions of doses of this. And the work to transfer them from making rabies vaccine to making our coronavirus vaccine based on rabies is a fairly small lift. And I really just wanted to emphasize that is how huge that is, given how fortunate we are here in the US and we are in Philadelphia working, for those of you listening who work for Jefferson, where we are in terms of getting our folks vaccinated. But really there's a lot of need throughout the rest of the world and a need to ramp things up quickly. So in addition to the other items on this list that Dr. Schnell went through, the fact that we expect long-term protection, safety for groups, and ease of storage and transportation, that's a very, very critical factor. I also want to build on what Heather Rose said about where we are. So we've licensed CoraVax for the developing world to a partner in India. That partner isn't going to make or sell vaccines in the US. And we're actively also, as she said, working to bring the coronavirus vaccine CoraVax to the US by evaluating and establishing a Jefferson-led process to do that. And sometimes people ask, why do that when there are other vaccines in the world? But this slide really helps cement that into place about why we think that is the right thing to be doing. We think that there's still a place for this vaccine in the world, and a very important place. We still think that when the story is told about how the world and the globe responded to this terrible pandemic, that there'll be a place for CoraVax, and there's a place and a name for Jefferson. I had one other slide, but I'm going to skip it in the interest of time because I really, really want to try to get some questions from people. So you can just go back to the group and go away from the slides for a moment, and we'll see what questions we have. So I'm going to turn it over to Rob. Thank you, Rob. Rob's going to, he's been looking at all the questions coming in on the chat, and he's going to moderate questions for us.
How are you, everyone? Thank you for joining us today. I'll start from the top. We've got a question from our very own Dr. Sindara, and I believe this is a question for Dr. Schnell. He's asking about your thoughts on oral vaccines, and he points out a new vaccine that, I guess, partnership that was announced between India, Israel, and an American company.
Well, that is a good question. It probably would be the best vaccine. if you have an oral or nasal vaccine, because that may be the only way to stop the initial infection, because you could get mucosal immunity. So I don't know the vaccine, but as I said, as better, and if it works, it certainly would help with a lot of things. You don't need a needle and so on. So basically, the holy grail of vaccine is an all vaccine with a single immunization, which is heat stable and cheap. If you put everything together, that would be, certainly something important to do. I mean, rabies vaccines, a live rabies vaccine is actually used widely as an oral vaccine to immunization of wildlife. That's how they eradicated fox rabies in Europe. But yeah, certainly makes sense.
Great, thank you very much. Moving on, this is, I think, something that a lot of people are talking about today regarding the variants that we're discovering with coronavirus. And this question comes, how does CoraVax, how could it respond? Could it respond to the emerging variants as we hear about them? And if not, what would we do to address variants?
I mean, for the variants, what everybody does right now, including us, is putting the variants in their vaccine. So the question is certainly, can you mix your vaccine? Can you make a polyvalent vaccine for rabies? For this vector, we showed actually that you can mix a vaccine against Ebola, Sudan, Marburg, and Lassa, inject that was done in non-human primate, and you get a response against all 4 antigens. So that's certainly the way to go. The other thing is, it has shown that the immune response is not getting totally lost. So as better your immune response is, as better probably you can protect. For example, the RNA vaccine, they do okay against the UK variant. The UK variant is really not a problem. People get it a little bit wrong. That's just a little bit of faster virus, so it spreads faster, but it's nicely neutralized by most vaccine. The variant in Brazil and South Africa is more of a problem. And South Africa is the worst by escape, but Brazil, I would be being closer, more concerned, and Brazil doesn't control their pandemic at all. It's almost crazy how the virus spread there. So that may be the first we get. So my point is, make new vaccine and mix them, right? The question is, what worries me is this old antigenic sin. If you get a vaccine, and that's shown for influenza, if you get a vaccine and then you boost with another vaccine which has some mutation, you often don't get a good response against the mutation, but just boost what your immune system already saw. So we have to see how that goes. And then certainly, what will be the side effect? RNA vaccine have some side effect after the second inoculation. You can get quite sick, and I was quite sick. What happens after the third one? Is it a platform or is it an antigen, right? Adenovirus, can you actually immunize multiple times? I think that's where the advantage comes to the vaccine like CoraVax or this VLP, which you don't have pre-existing immunity because they don't have to infect you. So we have to see. Right now, it's everybody should try everything, right? We don't know what the mutants will play for a role.
I thought maybe Dr.
Just-
Kuczynski would have a point around this too. Yeah. Maybe
Yeah, I was just going to sort of emphasize Matthias's point in another way. In answering the question, there's the tortoise and the hare here. Operation Warp Speed, made their bet on these pioneering, unprecedented vaccines, the RNA vaccines, the adenoviral vaccines, and so they've moved more quickly. And technically, there's no reason this vaccine couldn't have moved as quickly. It's just that there were more resources poured into it, and that's how it moved. And I think the question that is implicit in Matthias's presentation and Heather and Rose is really, why would we continue as the hare, as the tortoise? But our feeling is that it's not just for, first of all, for SARS-CoV-2, there are 8.2 billion people on planet Earth. 7.8 billion people on planet Earth, most of whom just simply will never ever be able to get those pioneering platforms because of affordability, transportability, et cetera. So there's that reason. And there's deep questions about revaccination on those other pioneering platforms in terms of their inflammatory properties, et cetera. So that's part of it. But then we're also thinking in the long game, really, because what about the next one, whether it's variants or an entirely different virus? What's going to be there for those vaccinations? And we do believe this is a very robust platform that can, in principle, give decades-long immunity, safe for pregnant women and children. And that's what's really driving Matthias and his team out of the vaccine center. So the more the merrier in our view. I think we're going to need lots of platforms on planet Earth to deal with these things. And we believe these kinds of viral vaccine platforms are quite robust and really will have a place now and into the future.
Great. Thank you very much, Dr. Tukachinsky and Dr. Schnell. I think we have time for one more. I think both, actually maybe two, because both of these can go to Dr. Rose. The first question was from Rob Arcatapane. You have a fascinating and incredible background. We're lucky to have you. You're almost the perfect person to be helping develop vaccine technology in a pandemic. What drew you out of the lab and into the business side of the equation? And then if you could follow that up with a question from Dr. Dicker, can you explain whether our patent for CoraVax was US or worldwide?
Right. Thanks for the kind question. So I will say that I consider development hand in hand with research. I think that what people like Dr. Ritz and I do is just as essential as what our research scientists are doing. I say this all the time. What we do doesn't matter unless it gets to the people who need it. And therefore, there has to be somebody on the other side of the continuum that thinks, what now, once it's done in the lab? And there's always a what now, almost always. I mean, it's rare that we have something that can go straight into a patient. And so I have a deal with myself that I would absolutely never take a job where science wasn't core to it because of how much I absolutely love the science part of what I do. But the only adaptation that I've made to myself in this role is that I'm just much more a generalist than I started out. Like I said, I get what I get. I don't get upset. I can't pick and choose day to day whether or not I get something I love working on or if it's totally new and I'm going to be learning something new. But that, to me, that's very thrilling. The patent was filed as a US provisional patent in the United States initially. That gives us 1 year runway to file something called a PCT patent, which stands for Patent Cooperation Treaty, PCT patent application. It reserves our right to file in any country around the world that's a PCT signatory. That's 153 different countries around the world that are PCT signatory countries. And it's most major jurisdictions where having a patent would matter. So there are some places where the countries may be honestly economically in need, having a patent wouldn't really be the barrier to getting technology to them. So, that does overlap well with who's not PCT signatory. So it's now currently in PCT stage.
So we're on track for full world coverage. Indeed.
All right, thank you so much for your questions. And I'll turn it over to Dr. Gentile O'Donnell to close this out for today.
Hi, everyone. Thank you all so much for being here. I think anyone who has taken the time to listen to Jefferson's rock stars will not be too disappointed that they haven't been to any rock concerts lately. There are really extraordinary things happening here, and these are the people that are making it happen. You may not be playing the air guitar, but one of these days, I think your names will all be up in lights for this remarkable work that you've done. So thank you all for being part of this. If anyone that you know was planning to join and couldn't, please know that this has been video recorded and is available on the Innovation website. So we will hope to see you soon. Thanks, everyone.
Thank you.